This shows the process of coagulation, and I want to focus on the bottom half of this diagram initially. Thromboxane is a chemical that's released with the injury, and then it attracts the platelets. Remember, I talked to you about the fact that the injury itself attracts the platelets to the site of the injury, and the chemical responsible for that, the chemotactic factor, if you will, is thromboxane. Now, the process of coagulation is where proteins in the blood attach to the platelets that have formed the plug, and this forms the full-fledged clot. Now, the process here is first the production of something called prothrombinase. You can see at the top on the right the term prothrombinase and the fact that it's associated with two factors, factor 10 and factor 5. We'll talk about this some more in just a minute. The second point is the fact that prothrombinase then converts prothrombin to thrombin, and you can see the formation of thrombin right in the middle of the diagram here. And thrombin actually has several functions, one of which is to continue with platelet activation, and then also the third step, which is to convert fibrinogen, which is that protein I told you floats around in the blood all the time, into fibrin. And when fibrin is produced, it attaches itself to the platelet plug and begins to form the full-fledged clot. And so we're looking at this section of the diagram at this point. Now we have the same diagram, but now we're going to move up to the top of the diagram. And the top of the diagram shows what's called coagulation factors. These are proteins in the blood that initiate coagulation. Hemophilia is a disease, an inherited deficiency of one of these factors. And each of these factors has been given a numerical name now. It used to be given a specific name. For example, factor 8 has this little VWF beside it. That stands for von Willebrand's factor. Factor 9 is also called Christmas factor. And the hemophilia, there's a von Willebrand's disease, which is associated with a factor 8 deficiency. There's classic hemophilia, which is also associated with a different kind of factor 8 deficiency. And then there's something called Christmas disease, which is associated with a factor 9 deficiency. But each of these factors come together to perform reactions that lead to the formation of the factor 10-5 complex, which leads to the production of prothrombinase, and then the latter part of the coagulation process. Now, the body also produces proteins, or has proteins present, that are responsible for stopping coagulation, so that once started, it doesn't cause us to just completely coagulate all of our blood. And these are called protein C and protein S. You don't need to know that. But protein C and protein S actually are activated during this process as well to stop and control how far along the coagulation process goes. So this time, talking about these upper factors is as indicated in the circle at this point. Now, diseases known as hemophilia are diseases in this section of the coagulation process. Now, let's move on and talk about blood types. In order to understand blood types, you have to understand a concept known as an antigen. An antigen is a protein or other molecule that triggers an antibody production by the body. And the antigens are almost always on the surface of cells. And these are the 4 different kinds of red blood cell blood types. And it shows that there are different antigens that are represented by each of the types. So blood cells that have antigen A on the surface, the blood is called type A, and anybody that doesn't have that will produce antibodies to type A. Blood type B is associated with a different antigen set, and produces different antibodies in people who don't have this. The AB people, their blood cells have both antigens on board. And then people who have type O are people that have neither of these major antigens. Now we'll talk in a minute about positive and negative, but these are called major factors or major blood groups. Now the universal donor is the person on the lower right here that has type O blood and has no antigens. Obviously, you could give this blood to someone else and they would not recognize it as foreign and would not react because there are no antigens to react to. The universal recipient is the person with the AB group because they've seen all the antigens before. They do not form antibodies to either. And so if you give them blood with any of these antigens on board, they will not react to it. Now the Rh factor is considered a minor protein, but it is still important in blood typing. And so a person either has the Rh antigen or they don't. If they have the Rh antigen, then they're called positive. If they do not have the Rh antigen, they're called negative. So you have O positive, O negative, AB positive, AB negative, and so forth. Now what I'm showing in this diagram is what can happen between mother and child in utero, depending on the child's blood type and the mother's blood type. If, as indicated on the left, the child is Rh positive and the mother is Rh negative, she recognizes the Rh antigen as a foreign antigen and she begins to form antibodies to that. Now this does not happen with the first child. Well, actually it does happen with the first child, but the antibodies don't become mature enough to attack the first child. So the attack doesn't occur until she has the next child. But during the process of delivery, some of the baby's blood leaks across and causes her to make antibodies. And once she makes antibodies, then the next child that has Rh positive, those antibodies can cross the placenta and attack the baby's blood cells. And this is what's known as hemolytic disease of the newborn. It's when antibodies to the Rh factor that are produced in the mother attack Rh positive baby blood cells. Now this obviously can only occur when the mother is Rh negative and the father is Rh positive, such that the baby has the opportunity to be Rh positive. And there's actually a way of preventing this, and it's called RhoGAM. You may or may not have heard of giving RhoGAM injections to women after they've had a baby. But what you do is if the mother is Rh negative and the baby is Rh positive, then you give the RhoGAM injection to the mother. And what the RhoGAM injection is antibodies to the positive antigen. And it binds all the positive antigens so that the mother doesn't see it. It masks the antigen and hides it from the mother so she doesn't see it and never forms antibodies to it. Now we're going to move on and talk about circulation. We're moving away now from talking about the blood, and we're now going to talk about how the blood gets around. And there's essentially in biology two types of circulation. There's open circulation and closed circulation. And we talked about this when we talked about mammals and lower life forms. Open or closed circulation is when the blood remains in a series of special tubes called blood vessels and never leaves those blood vessels. And so that it's closed in the fact that the blood is closed within a special system and never comes out of that special system. This is opposed to an open circulatory system in which at some point in its circulation, the blood leaves the circulatory system and mixes with the tissues, and then in some way is collected back into the circulatory system to go through again. Now, humans obviously have a closed circulatory system, and what we will be talking about from this point forward is closed circulation. Now, when we talk about circulation, we're talking about blood in blood vessels. So what are those blood vessels? Well, there are arteries, which are blood vessels. They are defined as blood vessels that carry blood away from the heart. Now, most of the time, this is oxygenated blood, but not always. So you do not define blood vessels by whether they're oxygenated blood or not. You define them whether they carry blood away from the heart or towards the heart. And arteries carry blood away from the heart. Veins, on the other hand, carry blood back to the heart or towards the heart. And then lastly, capillaries are microscopic blood vessels that connect the arteries to the veins. And they also are the place where gas exchange occurs and nutrient exchange occurs between the cells of the blood and the cells of the body. Now, when we talk about circulation, we're talking about two circulatory systems. There is the pulmonary circuit, which is the circulation of the blood through the lungs, and it is primarily shown in the top half of this diagram. You see that blood comes out of the right ventricle, and we'll get to more details about this in the future slides, heads out the pulmonary arteries, which are blue because they're carrying deoxygenated blood, into the lungs. They go through the capillaries of the lungs, and oxygen exchange occurs, and carbon dioxide is released. Then it comes back to the heart in pulmonary veins and into the left side of the heart, where it is pumped out into the second circulation, which is the systemic circulation. The systemic circulation circulates blood through the entire body. And what you see is once the blood enters the left side of the heart and goes out through the aorta, it then travels through the systemic arteries and into the body, the systemic capillary beds. Then it's recollected and comes back up through the veins and back into the right side of the heart and into the right ventricle. Now, essentially, the body is set up with two circulations, a circulation through the lungs and a circulation through the body. And this is necessary and is not seen in some of the lower life forms because we breathe air and we have a closed circulatory system. And so we have to have a separate circuit through the lungs to have things exchanged in the lungs. And it's separate from the exchange that occurs within the peripheral system.